Welcome back, everyone. After we've been through some more technical sessions about Julia and Neuroblox and how to code in both, now we'll move more into the neuroscience, and we'll start with some basic units of neuroscience, talking about neurons, neural mass models, and sources. And these three categories basically are the outline for this morning's lecture. So first, we're going to go through neurons, how their biology, and then how we represent them in computational models. Same for neural masses, what assumptions we make when we create neural masses. And then we'll talk about sources. And for sources, we have plenty of options in Neuroblocks. We have some simple sources, for instance, sinusoidal waves that you can input to any blocks, or some spike trains like Poisson series. But for now, I would like to focus on deep brain stimulation, because that's one category of sources that is very much a real world application, and we include this already in Neuroblocks. So moving on to neurons first. As you might know, our brain is full of them. Human brains can have around 10 to the 10 neurons. They're sparsely connected with each other, but still dendrites of most of these neurons receive on average an input from 10 to the 5 other neurons. And also neurons have a soma and an axon coming out of the soma. And the axon is how they communicate the axon potential to the dendrites of other neurons. And here on the right, you can see two images of neurons. On the far right, we have some pyramidal neuron that has been actually GFP labeled. You can see in green. And on the left, you can see a drawing that Ramon Cajal did of Purkinje cells and granule cells in the cerebellar cortex. So now, axon potentials are how neurons communicate with each other. These are chemical signals that neurons turn into electrical ones. And the way that this is done is there is some influx of ions from outside on the synaptic cleft to the inside, which is the soma of the neuron. Eventually, the soma of the neuron starts from the dendrites. And this influx happens through receptors, right? That the gates that essentially open at different times. And the first computational model that actually described these dynamics, these receptor dynamics in detail, was the famous Hatchkin-Huxley model. And we'll see different flavors of this model implemented in Neuroblocks. And another success of this model is essentially because of the way that its equations were laid out. Yes, it did describe the receptor dynamics, but it also offered us something else, which is an equivalent electrical circuit. So this is another way of thinking about the operation of single neurons, right? So we can create an electrical circuit with actual electrical components like receptors and capacitors and battery sources, and a circuit like this would behave in equivalence to an actual neuron. So this is a nice both abstract but also more practical at the same time representation of this computational model. So in more detail, the synaptic transmission. As I said, this is a chemical signal. We have synaptic vesicles being packaged on the presynaptic side, and then they get emitted in the synaptic cleft, and they're picked up by receptors of the postsynaptic neuron, which consequently the receptors actually open the ion channels, and we have the ion influx that actually causes the action of the postsynaptic potential, because the influx actually potentiates the neuron. And people have observed these two types of neurons. We call integrators in class 1 and resonators class 2. So we can distinguish in them by their Fi curve, which is the firing rate frequency over the injected currents to the neuron, right? So we have neurons that actually, as you inject more and more current, right, the firing rate goes up almost proportionally to the injected current. But also we have the resonators, which you can think of, their function almost like a switch. So up to some specific input current level, nothing happens, there's no firing, and then there's a jump to some firing rate, which might grow proportionally afterwards for higher values. And here on the right, I just wanted to show you some face portraits of how this is possible, and this coming from a book by Idzikiewicz. This is the same Idzikiewicz whose model you have implemented, and actually that model can exhibit these bifurcations. And this by bifurcation, essentially we mean different configurations of the variables and different evolutions of the combination of variables that allow for either spiking or silence, or moving between rhythmic spiking to silence, or the other way around, or moving between bursts to single spike, and so on. And of course, to draw diagrams like these is much easier for simple neuron models like the Idzikiewicz neuron, which is only two-dimensional. We can actually do this. It's computationally much easier compared to doing this for Hatching-Huxley neurons who have more equations. So you need to actually explore a larger parameter set and do this in a simulation-based manner. So this is also a comment towards the benefits of simple neuronal models, right? You have a simple model, maybe the parameters and the variables that it carries, like the Idzikiewicz neuron, they are less biologically relevant, but you know more about the behavior that that model can exhibit more easily, sometimes even in paper. Right, moving on to the second category, these are the neural mass models. So you can think of neural mass models as a step up on the abstraction ladder. Such models were designed to capture, on average, the activity of neurons connected to one another, and in general, they are oscillators or collections of oscillators. And here you can see an example of that, which is the Kuramoto oscillator. So these are very much phenomenological models, as in they don't rely on biology. They just imitate, this periodicity in spiking, right? But in very simple terms. So they just assume this sinusoidal spiking, and have a pool of them. But the handy thing about the Kuramoto oscillators, as you can see in the diagram, is that given a pool of oscillators, you can, by changing this one variable in the model, you can move from regimes of, all of the oscillators oscillating in a similar frequency. So now you have synchronicity, to regimes where oscillators are more, uniformly scattered and there is no synchronicity, or they're split into different clusters. So you have some flexibility in this model. Now, there are even more detailed neural mass models that allow for more flexibility. And again, these are all examples that we have already included in NeuroBlox, like the Wilson-Cohen model, which is a model that describes an EI balance in a neuronal population. The Janssen-Reed model, that is again a model that describes interaction between pyramidal neurons and interneurons. Or Latter-Breakspear, which you can actually see a face portrait of that here on the right. And I'm showing you the face portrait so you can see actually the flexibility in this model, right? So each color here is a different regime that the model can be in, depending on its parameter values. So you can actually tune it and observe different behaviors in this model, according to your data as well. So there's even more flexibility to the simple Kuramoto oscillators from before. And for maximal flexibility, I would say, we have these next generation neural mass models, again, already implemented in NeuroBlox. So these models are maximally flexible because they're analytically derived from a population of neuronal models, right? So we first assume how are neurons behaving. For instance, they have been already analytically derived in the literature for QIF neurons. That's the quadratic integrated firing neurons you have seen in previous exercises, or Idzikiewicz neurons that you have implemented yourselves. So if you assume a population of such neurons and some connectivity between them, you can analytically derive a next generation neural mass model that is more flexible than the previous models that I've shown you. And you can see an example of this flexibility here. For instance, this period of synchronous activity, synchronous spiking in our population is actually longer than the quiescence period and the other activity periods, right? So you can even tell from this image that this model can do more than simple periodic activity, right? It doesn't assume sinusoidal neurons inside it. It's more flexible, so you can capture even transit periods like that, or shorter or longer periods of activity and quiescence. So the final section that I wanted to talk about is our sources, and specifically deep brain stimulation. So deep brain stimulation is a neurosurgical procedure. It has been applied. I think one classic application of it is Parkinson's disease to reduce motor symptoms like tremor and rigidity. The benefits of it is that it is adjustable and reversible, and it allows thus for better medication management. But also now it has been extended, and other brain disorders can also be treated with DBS. So as I was saying, this is very much a real-world application, and we're trying to match exactly those real-world protocols in NeuroBlox. So these are two examples of stimuli from the DBS suite of stimuli that we have. For instance, you can use this DBS simple structure given all these parameters with the relevant units, which will essentially create a source that operates like this. You have square pulses interspersed in some periodic manner, and here you can see just a zoomed in image of a squared pulse. You can have it with smoothed corners or completely square. But also we offer more flexibility, so you have these more complex protocols where you have like a burst of pulses, and then a period of silence, and then a burst again. So this is the protocol DBS over the previous structure that was just DBS. And finally, I just want to mention that we implemented those structures, both of DBS, and we replicated results from a deep brain stimulation paper that essentially modeling DBS in basal ganglia, and we have a model of this, and these are just power spectrum results from simulating the model in these three regimes under different stimulation conditions. And you can see more about this in the exercises as well. So this was my last slide. So similar to previous days, you can navigate to the neural masses and sources day. This is the web page for the session, and you can work on the neural mass notebook. So yeah, and feel free to ask any questions anytime. Thank you.